Welcome to our series, Microorganisms Through a Different Lens. It's a part of the Masters of Beautiful Achievement series, a place where I investigate systemic, sustainable solutions using nature as our inspiration. The Microorganisms Through a Different Lens is hosted by Mark van Bemmel and Alexander Prince, and each month we invite a guest in the field of virology, microbiology, and biochemistry for an explorative dialogue on how we look at our natural world to discover how this impacts on how we look at life sciences, biotechnology, and environmental sciences. And in today's episode, we will explore our gut microbial system, about the how and the why it's affecting our health systems. We are curious to learn what kind of insights this will provide us for our understanding of microbial systems, and how do these microbial metabolic pathways in the oxygen-free environments work. And of course, Mark and Nick are very curious what kind of interesting applications we can extract from this. First, let us introduce ourselves. My name is Alexander Princeton, and with my company Scope Matters, we operate on the cross-section where technology, product innovation, and material resources intersect. We help businesses to apply ecological thinking to bridge economic doing to ensure circularity is the only logic outcome. And over to you, Mark.
Yes, I'm Mark van Bemmel. I'm a CEO of Orvion. Orvion is a biotech company focusing on water and environment. And we study microbes a lot in our laboratory with sequencing techniques and PCR techniques. And yeah, I'm quite interested to hear about the gut microbiome, which I do not know so much of, but I've seen a lot of microbiomes from soils and water systems, so there's probably a lot to learn from each other.
And today's special guest is Koen Venema from Maastricht University. Please, Koen, please introduce yourselves to us.
Yes, well, first of all, thank you very much for the invitation for this podcast. And I really hope to surprise you and the listeners about the gut microbiota tonight. So I am a professor at Maastricht University, and I have a so-called chair in gut microbiology. And on top of that, I am a founder and CEO of a consultancy company on beneficial microbes. And it is particularly the health benefits that the microorganisms in our gut can provide that makes it very interesting for me to study them.
And thank you. And we invited you, Koen, to explain us a little bit what's happening in our gut system. Everybody knows, or at least most people should know, that our gut system is full of microbes and bacteria and fungi. And yet we are slowly discovering that they actually have a very important function to play in our health and even in the way we think. First of all, maybe it's interesting to note, how did you end up in this field, Koen?
Well, that goes back to high school when I was interested in biochemistry. So I started studying biochemistry at the University of Groningen, and then did a topic at the molecular genetics department where they worked on lactic acid bacteria that are used in food fermentation, so production of cheese or fermented sausage or sauerkraut. And slowly I rolled into then gut microbiology, because also these lactic acid bacteria are currently used as beneficial microbes that you can eat, and you become supposedly more healthy from that. An example is, for instance, the bacteria that are present in this little Yakult drink that you can buy in the supermarket.
But you mentioned supposedly.
Yes, there is a lot to talk about that. I can fill 3 hours with that, and I won't do that here now. But it really depends on which microbe you ingest, whether or not you actually have a benefit for what you want to have a benefit for. For instance, if you want to have a different stool transit, you should take a particular bacteria, but not a random other one, because that won't help on stool transit. The other one may help on allergy.
What is stool transit?
So if you're constipated or if you have diarrhea, yeah. So it can normalize that back to a normal stool transit.
So you don't have to eat bananas or rice or.
That's of course also a possibility, and then you ask your own microorganisms in your own gut to help you with your stool. But you can also take a what is called a probiotic, right, to modulate your gut microbiota or your stool transit or your skin allergy or even things related to the brain, such as cognition or brain development in young children. Microbes are even play a role in that.
And also for the listeners, just to frame it, the research in gut microbiology is quite new if you compare it to the other fields, isn't it?
It's indeed relatively new, and that is because most of these microbes that are present there cannot be cultured by us as single isolates in the laboratory. Simply because collectively in the world we are not clever enough to know what they need to grow. And if you imagine that there are 10 to the power 14 microbes in our guts, or 100 times a million times a million, in fact, there are more microbes in our gut than we have cells, but because they are microorganisms, they actually fit in there. Because we can't culture them, it limited the research that we could do on these microorganisms. But with next generation sequencing technology, we now can identify the microorganisms that are present there. And that has really revolutionized the field in gut microbiology.
Yes. That's an extremely interesting topic, of course, because we also do a lot of NGS in our laboratory, and we look at complex and biodiverse microbiomes. In fact, I'm quite interested to know, is there a relationship between the diversity of the microbiome, I mean the number of species, and your health? Is that something that has been found, or?
That is something that has been found for certain diseases, yes.
Yeah.
But you should really be careful about that, because, for instance, when we get older, also our diversity changes, and of course we can get old while not being healthy, but there are also a lot of healthy older people. There are a lot of centenarians that are also still healthy, and also their microbiome changes, simply because we as a host change, and these microorganisms that reside inside us, they react to those changes. Yes. So diversity has been shown to matter in a number of diseases, but is not the single parameter that explains everything.
No, obviously. So, but if you get a severe disbalance, something like one microbe gets very dominant, this could lead to disease or to problems. Is that something you can say in general, or?
That used to be Koch's postulate, right? So a single microorganism is responsible for a certain disease. Right. With gut microbiology, that is very difficult to conclude. Each person has approximately 250 to perhaps 500 different microorganisms in the gut.
Yeah,
but they can do the same trick, right? And each person has its own composition of the gut microbiota that can be sourced from perhaps up to 5,000 different microorganisms that can be present. And because of next generation sequencing, we know that people that are analyzed all over the world, they have a completely different gut microbiota.
Different even.
Well, yes. There's been studies that talked about the core microbiota, meaning microbes that are present in everybody. Yeah. That was studied with, I think, 121 individuals, and there they found that if you take that 100% level really being present in everybody, there were only 17 or 18 microbes that were present in 121 individuals.
Oh, wow.
Okay.
Now we expand that number of people, we slowly go down to 0 microorganisms that are present in everybody.
Oh, really?
Well, that is
So
weird.
probably, I think that it's in your presentation, but it can also change over time. So as a baby, you have a completely different population as later in life, and maybe it changes all over your lifetime.
It does, yes.
Yeah.
We consider it to be relatively stable during adulthood.
Okay.
But of course, there can be perturbations like taking a course of antibiotics. If you're sick, right, and you need it, then it doesn't only affect the pathogen, but it also affects other microorganisms in your gut microbiota. So you will really disturb it. And when we are born, we are basically born sterile. Yeah. Although there is a discussion about that, but for the sake of tonight, we'll say we're born sterile, and then slowly you get populated with microorganisms. And it really depends then, for instance, if the baby gets breastfed or formula fed. In breast milk, there are microorganisms, so they are transferred from the mother to the baby. And in a sterile formula bottle, there are, of course, no microbes. So there's already a big difference there.
That's very interesting. Could it even be that this changes your microbiome for your lifetime?
There are ideas indeed that may be the case, or if not your microbiome, then certainly your own metabolism, because these microbes interact with us as a host, right? And they influence our immune system, they influence our metabolism and even our brain.
Yeah, I mean, I know about the last one is that these bacteria have their own pathway to our brain next to our nervous system, which is quite weird if you consider that is what you eat is what you are, literally. Yeah. But the microbes are the ones who are translated that.
But that's extremely interesting because the use of the change from breastfeeding to bottle feeding, how do you say, has been quite abrupt, I think, in history. Dates back to the 70s or 60s, maybe. Can you see a shift leading to, for instance, certain diseases that can be linked to that? Or it's very
it's difficult to say because there's been other changes as well, right?
Yes.
Over time, we've become much more hygienic than we used to be.
Yes.
And there is a hypothesis that staying away from nature actually also influences our health.
Yeah.
And influences our gut microbiome as well. So I've got 5 kids. 3 are adults and 2 are quite young. The last 2 we can just let them play in the garden, and if they want to eat some soil, then we'll let them eat soil. But the first 3 no, because there the idea was that's bad for you, but in fact it's actually good for you.
It's actually good, yes.
Yeah, because I did some research on the microbes you have been discovering, and I have Actinobacteria, Bacteroidetes, Firmicutes. Those names are always challenging. Proteobacterium, and then you have Vericumicrobia, and Fusobacterium. And what I found is that they're all linked to soil and fresh water and aquatic and terra environments.
Yes, these are names at what we call a very high level of phylogeny, of classification of bacteria. There's 6 or 7 or 8 levels of classification that you can use, and these are quite high. But it does mean that all bacteria almost fall into those categories, and yes, you do find proteobacteria in our gut and also in soil and in water and what have you. And in fact, this may be one of the ways on which we get colonized by these bacteria. We've recently found out that within the fruit and vegetables, so not on the surface, but actually within the fruit or within the leaves of vegetables, there are bacteria. So certainly if you eat those raw, those bacteria will enter your gastrointestinal tract and may actually also establish there and become part of your gut microbiota. Yeah. This is a field of research that we are currently engaged in, and we hope to be at the forefront there to discover these kinds of correlations.
But then you're also suggesting that the fruits and vegetables coming out of greenhouses, they tend to be more sterile than the one grown on the soil.
Partly, but definitely there's a difference in this diversity that Mark mentioned earlier, the number of different microorganisms, which in a greenhouse is much lower than in a organic or biologically grown crop. Yes. Yes. And we think that diversity actually matters, and that a diverse microbiome may be more healthy for you than one that is less diverse.
That's interesting. So food is our medicine, but the food includes microbes.
Well, of course, yes.
Yeah, no, but we're all trained like food is our medicine, and then we eat food, and then we don't think about the smaller creatures. But actually, what you mentioned, Koen, which challenge I think most, even the growers I work with in here in the Westland, you're challenging their beliefs, because a vegetable includes microbes, and that is the medicine, not just the thing that we can see.
No, but also we tend to be to wash our green stuff and so on very, very carefully and very thoroughly. But actually, it should maybe a bit better to keep some of the garden on it. Or
yeah, no, I agree, and you know, in industrial practices, they are washed with some chlorine even, although in very low concentrations, but to make them more clean and more sterile. But yeah, perhaps we shouldn't do that.
Or add probiotic.
Yeah, or I was going to say, or if you buy them from the supermarket, just drop them on the floor and then eat them or something like that.
But there is the 3-second rule, isn't it?
Oh yes.
But it's interesting what you're saying, Koen, is that, I mean, supermarkets should then sort of like probiotically enhance their vegetables again with the species that should be on there, and then sell that as a whole package.
Yeah, so there is a difference between the species that are on there and species that are in there.
Yeah, okay.
So during growth of a crop, these microbes enter the leaves and eventually the fruit, and they are even present on seeds. So even from plant to plant, through the seeds, those microbes may be transferred and may be involved in health of the crop. And of course, we know that very well for the microbiota present around the roots of crop that help the plants to grow and catch nitrogen. Yes. The rhizobial microbiota. Yeah. But we also now think that perhaps in other parts of the plant, the microbiota is essential for health of the plant.
Yeah. Would you say these rhizobial bacteria are also important for your gut? So is that known? Or?
Who knows? I never given that much thought, frankly. But it might very well be. But if you look at all the different microbes that are present there, right? The idea is that their metabolic capacity, the things that they can do, outnumbers the capacity of our own liver. So they really can do a lot of different things.
Oh, wow.
Yeah. The difference between the liver and your gut microbiota is oxygen.
Yeah.
Right? There's no oxygen in your gut. So they have to do their thing, so to say, through fermentation, a breakdown in the absence of oxygen.
So maybe you could say that this bacterial diversity in your intestines, in your gut, it just increases the metabolic capacity of your entire being a lot by an enormous factor, because all these different bacterial strains have completely different metabolic routes and possibilities.
Yes. Yeah. Yes, a good example for that would be to look at carbohydrates that we ingest.
Yes.
Our own body can only absorb and digest a limited number of sugars.
Yes.
Free glucose, free fructose, lactose from dairy products that is digested, and starch,
Right.
which is converted into glucose. If you look at the number of carbohydrates that we ingest normally on a healthy diet, there are hundreds of complex carbohydrates that we cannot use ourselves.
Right.
We rely on our gut microbiota to digest those carbohydrates, to cut them into pieces.
Yeah.
And they have, depending on the microbe that you look at, they have between 30 and perhaps 200 different enzymes to break down those complex carbohydrates and make them available for us through fermentation.
Yeah.
And but then I have the question: and what does our tummy play a role in it? Because that's the most acid place before it gets into the intestines.
That's actually an interesting question, because that's one of our defense mechanisms, right? Gastric acid. And the idea is that is one of the barriers for microbes to enter our body, because they are killed by the high concentrations of acid in our stomach. And yet, if you look at the immense numbers of microbes that are present in our gut, that's the only routes through which they could have gotten there, through oral ingestion.
Right.
Now, what we do know is that very young infants, they do not secrete as much gastric acid as adults do. So their gastric pH is not very low, so that allows for survival of some microorganisms that are ingested. But also transit of the meal from the stomach to the small intestine actually matters there. It takes about 3 hours after a meal, and then your stomach is empty again. And if those microbes are transferred from your stomach to the small intestine in the first half an hour, say, then your pH in the stomach is not as low yet, because the meal that you took has a buffering capacity, and that allows live microorganisms actually to enter the small intestine.
Yeah.
Now there they actually get hit on the head with a second defense mechanism, bile, which breaks them open, lyses them, and kills them essentially. So only those microorganisms that can stand that second hit with the hammer, so to speak, they will make it to your large intestine and are able to establish there.
Yeah.
So the pathogens are, when they come through, they are smart as hell.
They are, yes.
Or they're with a lot. It probably has to do with the dose as well. It will make a difference if you take 1,000 or 1 billion of pathogenic bacteria, probably.
Yeah, that's salmonella, for instance. When you're unlucky, it gets through the system.
Exactly. Yeah.
Yes, and listeria, same thing. A few cells can get you very sick if they are alive enough to interact with your epithelial cells or your immune cells, because then they are inside you, and then there's almost no stopping them, or at least no quick way, because your immune system needs time to respond to them.
But you also don't have bacteria or the bacteriophages, the ones like Mark is a fan of. Do you have them also in your intestine system as a protection to the invaders, or as a natural check and balance?
It's a natural check and balance. Yeah, we've talked so far about bacteria, but there are fungi, yeasts, viruses, bacteriophages, even some parasites, which are all considered to be microorganisms. And the interesting thing is that they all interact with one another. These bacteriophages, in fact, are viruses for bacteria. But other viruses affect the fungal and yeast populations, and they all essentially want to eat whatever we leave over from our meal, right? Whatever makes it to the large intestine, they want to have a feast on that. And so there's a lot of competition for these nutrients. And if you can what's the word I'm looking for? If you can benefit better from certain substrates than your neighbor, then of course you are in advantage. And but if your neighbor produces a antimicrobial compound that kills you, then of course you are again at a disadvantage. So there's a lot of warfare going on in your gut without you noticing it.
Right. What I find also very interesting topic is the exchange of information in the by exosomes and all kinds of DNA or RNA exchanges, maybe by viruses as well. Is it something that is important in your gut microbiome, you think?
Yes, we do believe so. And most of the research being done there is with respect to antibiotic resistance genes.
Right.
Let's take a hypothetical situation that a probiotic is resistant against a certain antibiotic, and you would ingest it, and the probiotic unwillingly transfers that gene to a pathogen. Yeah. And suddenly that pathogen becomes resistant to the antibiotic.
Yes. Yeah.
It happens. Yes.
Yeah.
Wow. Well, that is sort of like profound in that way. I mean, if they can hamper with antibiotics in a way they can exchange it, then they are like a smart brain fighting against their own extinction.
Yes. Yeah, they are very good at that. They can adapt very quickly to changes in environments, much quicker than we can, of course. I mean, we need thousands of years or even longer to adapt to changes in environments. Bacteria can do that in several hours.
Yeah. And in what way have humans interacted with the DNA of our gut microbiome?
I wanted to ask that question.
Well, in a certain way, I mean, these microbes were there before we were there, and that's clear. But essentially, we carry remnants of microbes with us in every cell. Mitochondria are believed to stem from microbes, and also chloroplasts in plants are supposed to be stemming from microbes. So, and during evolution, that has changed, and they got their own specific function. But I think without microbes, we wouldn't exist. But the other way around is also true. You know, the gut microbiota would not exist without us. We are a vessel, a fermenter, if you like, for these gut microbes.
Yeah, and what kind of metabolic pathways do you have? We talked about the Amorax bacterium and for the ammonia and nitrate pathways. I mean, hydrogen is almost always found somewhere.
Yeah.
Do you see the same traits in our gut system, and they exchange these gases or even the acids with each other as feast and feed?
They do, yes. It's something that we call cross-feeding, where one microorganism produces something that another one uses or changes, and that happens indeed in some acids that are produced from carbohydrate fermentation. It also happens with gases that are produced. Roughly a third of the world population, no, I should rephrase that. Roughly a third of the microbiota of the world population can produce methane. So they contribute to our problems in ozone layer and stuff like that. But we don't know why the other 2/3 don't. But the methane is produced by the gut microbiota by taking CO2 gas and hydrogen and coupling that together to methane.
Which is logical, because CO2 is most of the time a waste product from a fermentation.
Yes. And by removing that, you drive the fermentation into the direction of better efficiency and more energy production. Yes.
Exactly.
But
the methane is not being converted again by another microbe to.
No, mostly that is expelled through the lungs.
Oh wow.
To air. Yes.
So the carbon is then a waste product, which together with hydrogen, which needs to be ejected from the system.
Yes. which essentially is carbon loss, right? Even for these microbes, because if there would have been a way to incorporate it into their own biomass, they would be able to generate more of themselves. But at a certain moment in those fermentation reactions, the efficiency of energy generation is so low that it's easier to get rid of the waste products than to convert them any further.
Exactly. Yeah. Another question is, you mentioned the carbohydrates that are in our food are very important, obviously, and they're cut down by the bacteria into stuff that we can absorb with the epithelial cells in our gut. But there must also be some kind of a competition, because these smaller molecules are also very interesting for other bacteria to eat, obviously.
Yes.
Yes. So how is that functioning? I mean, how does our gut take up the goodies in a way before there are before other bacteria eat them?
Yeah. So I may have posted a little bit too simple earlier, but they do cut the carbohydrates in pieces. And that primarily happens on the outside of these microbial cells, because the polymers are too big to ingest within these cells. But then those sugars are then absorbed by the microbes and fermented, and they are fermented into what is called short-chain fatty acids.
Yes.
These are acetic acid, propionic acid, butyric acid primarily. Acetic acid is something that we sprinkle over our salads, so we even eat that. Propionic acid is also present in some foods, and is used as a conservative substance. Butyric acid or butyrate is not present in food, because if it would be, you wouldn't eat it, because it smells horribly.
It smells horribly, yeah.
It's incredible, yes. But these acids, they are absorbed by our body, and they are used as an energy substrate, a very important energy substrate, in fact, particularly for the epithelial cells that use approximately 70% of their fuel from microbially produced butyrate.
Oh, wow.
Right.
If that would not be there, you would have holes in your epithelial layer. Not literally, but the barrier function would definitely be disturbed because these cells do not get enough fuel, do not get enough energy to function.
Grow.
Yeah.
They grow fast, isn't it? The epithelial cells, they renew much faster than other cells.
Yeah, they say it's about a steak a week or something like that. Yes, it's a lot.
Yeah. And those are also the cells that are infested by the coronavirus, is it?
Not only those, but yes, those are also infested. Yeah,
that's interesting. So, are these epithelial cells just much better in taking up these fatty acids, or than other bacteria are, or is it some kind of take-up mechanism that makes them efficient for the body to?
Yeah, there is competition going on, because, for instance, acetic acid or acetate, in fact, with the pH in the gut, can be used by other microbes to generate more energy out of it, and then they can, for instance, couple two acetate molecules together to butyrate. But the majority of what is produced by these gut microbes, as the gases that we just discussed, is considered waste material. So it's excreted by these microbes in the lumen of the gut, and then it can diffuse to our own epithelial layer, and then is absorbed, eventually reaches the portal vein, is transported to the liver, and used there for energy, or if it's not absorbed by the liver, it travels to your muscles and brain and is used there as energy. So
wow.
We really need those microbes.
And we're still talking about, among others, butyrate.
Yes.
It's amazing. I'm just googling it while you guys were talking, and it's actually butyrate is also for an industry a drop-in chemical to make coatings and paints and other products. So it's interesting to see that actually, I mean, the difficult word you mentioned of the thing that was eating butyrate, but that is a sort of the coating of our gut system.
Yes, that's the single cell layer that divides the inside of our body from the outside. Because essentially our gut is the outside, right? There's a hollow pipe from your mouth to the other end, which is in contact with the outside world, essentially. So that single cell layer is the protective barrier that prevents the outside from entering our body. But yet it uses some of these waste products of these microbes to function properly and to function optimally.
And Mark, we have.
Sorry.
Yeah, go ahead, and I'll throw it in.
So this butyrate is very important for the health of the epithelial cells of this barrier. Is that right?
Correct. Yes.
Yeah. Okay, and so yeah.
If you remember your name, I'll jump in, because we had Mark van Loosdrecht, and with Mark we talked about biofilms.
Yeah.
And microbial species form together a community in which they strengthen themselves, and then the biofilm gets more, let's say, strengthened to ensure that the community grows. The way you're talking about this, Koen, it seems to be very similar to that.
Well, they at least they do interact with one another, and they send signals to one another, and they seem to sense with how many of them they are out there because of some of the molecules that they produce and that are seen. I mean, they don't have eyes, of course, but they are sensed by receptors on the cell surface. So they do communicate with one another. And I mean, I talked about warfare before, but all those reactions that occur by cross-feeding, where they supply food, if you like, for others, that's of course not warfare. That's the opposite. That's, helping your neighbors. And that is why we consider that community that is present there to be relatively stable over adulthood, because they do interact a lot with one another, in addition to interacting with us as a host. Yes.
Yeah. Okay, and could you? What I find interesting is what happens when the pathogen enters in this a good functioning microbiome gut, and is it solely the body that tries to get rid of this invasion, or is it also the microbiome itself, the gut microbiota itself, that will fight the intruders, or how would you say that?
Yes.
Yeah.
Yeah, the latter definitely also is the case,
Right.
because they compete with this pathogen for nutrients. So if they are very efficient in that, then the pathogen will not get any food and will not become active. They compete with the pathogen for adherence sites. Pathogens usually need to enter the body, and for that they need to first attach to the body to be able to start mechanisms to enter it. And if there are already other microorganisms that occupy that attachment site, then the pathogens cannot adhere. They produce compounds like these acids that we talk about that inhibit pathogens. So there's a number of mechanisms by which our own gut microbiota protect us against pathogens that we might ingest one way or another. Yes.
And the healthier the flora, the better protection.
Yes.
Yes.
So I mean, if we Westerns go to India, I think the first week we need to adapt to the new microbial flora that exists in India.
Yes.
Yes.
I mean,
Africa, yeah.
I mean, this is sort of like you're explaining, is that if you're in a different environment and your intake is different, then your gut system needs to adapt.
Yes.
To the I'm curious, in your research, have you ever done the gut research from a Western living in the Western world, and then he immigrates or moves into other regions where they have a different microbial flora, and then you can actually see the microbial flora changing to the local culture.
I haven't done that personally, but there's data out there where they have done that, specifically with Japanese people that went and worked in the US. Now, part of that may be because they also adapt a different dietary intake, and diet dramatically influences your gut microbiota composition, but it also is indeed that they are exposed to other environmental microorganisms and things like that. So yes, their gut microbiota does change.
Yeah. And now, of course, the holy grail is especially for people who are ill or more vulnerable. So how can you get a good microbiome?
Yes,
and that's what your research is focusing on to some extent, I think. Or
it is, and we use sophisticated in vitro models for that mimic the gastrointestinal tract that have been developed by TNO in the past.
Yeah,
and these are so good that they really mimic properly what happens in your own gut, and we use those to study what happens if you ingest particular food components and how that changes your gut microbiota, both in composition, but also in then what they produce and what we use again as substrate. So far, we've actually talked about the beneficial side of it, but when they ferment protein, they actually produce all kinds of toxic metabolites.
Right.
So they're not only very good for us, it really depends on what we eat. They can also do detrimental effects. They can produce ammonia or hydrogen sulfide, another one of those very smelly metabolites that are actually toxic. Hydrogen sulfide at high concentrations for our neurons.
Well, we have to eat a lot of protein because that's good for us. At least that's being told.
Yeah, a lot of protein, but not too much. And our body is very good at digesting animal protein and less so plant protein.
Oh wow! But that is a Western diet or generic globally?
That's essentially animal protein is generic globally. I should say that with plant protein, it matters whether you ingest the pure protein, which sometimes happens with supplements, or whether you ingest the actual whole plant. Because if you ingest plants, you also ingest fiber. And microbes in your gut prefer to ferment the fiber rather than the proteins, because they would like to use the proteins to build into their own enzyme machinery and to multiply themselves. So if there's fiber present, then that's beneficial.
But then again, if you take that to the next level, Asia is a continent which understands the benefit of fermenting food. I mean,
yes.
The Western world is used to processed food, and the East uses fermentation to process our food. Which provides a completely different end compound. What can you then say about fermented food? I mean, you can ferment protein, and that changes the whole composition, and also basically you could say the acid structure you eat.
Yeah. So most of the fermented foods end up being acidified. Sauerkraut is acidic, right? Cheese is cheese because the milk becomes acidified, and that clots the protein in milk, and then you get cheese. Buttermilk is acid. So all fermented foods become acidified, and the protein that is present in those foods to a certain extent become incorporated into microbial biomass. And in some cases, those microbes are actually helping digesting some of our food components. I earlier mentioned lactose as one of the sugars that we can digest. Well, actually, that's only true for the Western population. In Asia and South America, most of the people cannot digest lactose. And actually, we in the Western world are the mutants, so to speak. not being able to digest lactose is the normal way.
Yeah.
But if they take lactose together with some of these microorganisms, particularly what is called lactic acid bacteria, these bacteria help in digestion of the lactose in your body, in your GI tract. So in that way, they can stand the lactose also and could ingest dairy products. So there's a lot of benefits for also fermented foods to take. And nowadays, most novel probiotic microorganisms that are discovered come from traditionally fermented foods.
Right. Okay, and people who eat that by tradition are more healthy because of that.
Yes, I would say so. Yes.
Yeah. That's interesting.
But then how is the breast milk then also more fermented? Or I mean, raw milk tends to have more microbial flora than the pasteurized or homogenized milk we consume.
Yes, and we do that to be able to store it for a longer period of time, right? But I mean, thousands of years ago, fermentation of milk was the way to actually store it, because we didn't have refrigerators then. So yes, fermentation also is a means of conservation.
Right.
But then if you take that one step further, then I mean, if you look at the current food industry, and I know, for instance, that Wageningen is pushing on more fermented components into food as an additive or preservative. Are we moving to? Would it be smart for our human microbiome to move more to fermented processed food than the way we're pasteurizing everything?
At least it would be good to move away from highly processed foods where you have simple sugars rather than the complex fibers. Because the complex fibers are the meal for your gut microbiota. The simple sugars are taken up by us and never reach the microbes. So they are left without their feast. They are left without food.
They have nothing to do. They get lazy.
No, and they will start to feast on one another because they've got nothing better to do. So that's probably also not what we want. But if you look at the percentage of the Dutch or any Western civilization population that reaches the recommended amount of dietary fiber, that's less than 20%. And the amounts are 30 grams for women and 35 for men, if I'm not mistaken. But we've once did a study with a tribe in Tanzania. And there they easily ingest more than 100 grams of dietary fiber per day, which is all substrate for your gut microbiota. So their microbes must be very happy there.
But does it then help to blend your food? I'm having fruit shakes every day because I have how do you call that? I have steel in my mouth. I don't know what the English word. Ik heb een beugel. So consuming tough hydrocarbons is difficult, so I'm putting everything in a blender. But I like to consume a lot of fruit, so I put a lot of fruit in the blender and put some blend also some fibers from muesli fibers. So, but is blending then good, or would it be better to have the bigger pieces?
So for the digestible fiber, it helps to get them digested because you break them up in smaller pieces. For the dietary fiber, for the fiber that we cannot digest ourselves, it doesn't really matter how big those pieces are, because compared to a microbe, which is just a few micron large, those pieces are still big.
Okay, but then I'm
bigger, it doesn't matter.
Then I'm rest assured.
Yes.
But then I'm sorry, Mark. How does our poo look if we have a good microbial flora? I mean, Mark, you're laughing, but I have I received a son 8 weeks ago, and then you go through the phases of the poo.
Oh yeah.
First it's black, and then it's green, and then slowly it moves to yellow.
Yes. And eventually will get brown. Yeah.
Yeah. And that is amazing. And it all has to do with also with the microbial flora of the child.
It does. Yes. I've been on TV with that in a children's program where the question was, why is poop brown? So I had to explain that, and it takes me an hour to do that, so I won't. But it does have to do with your gut microbiota, indeed. Yes.
Yeah.
And I've learned. I mean, if you know how your poo looks like, you know how if you're healthy or not. That's also sort of like an indication.
Yes, it is. There is a chart, a scale for what your poop looks like. And you should be at a certain level to have a healthy poop. Yes.
Ah, that's very simple. You can monitor it yourself.
You can. Yeah. It's called the Bristol Stool Scale. Yes.
Just in general, maybe for the people who listen to this podcast, and you can buy all kinds of probiotic everywhere.
Yes.
Is there something to say about how much of it is nonsense and how much of it is actually does have some benefit? It's a difficult question, I know, Koen, but
well, it's not so much of a difficult question. I'm just trying to figure out where to start.
Yeah.
I mean, if all is well, you should not be able to buy a product on the Dutch market that has the word probiotic on it. No. Because the European Food and Safety Authority ruled against that. They say there's not enough evidence for a probiotic to be called a probiotic. And that is because in the definition of probiotic, implicitly, there is a health claim. So the European Food Safety Authority does not allow that. But it is very difficult to police that, to maintain that. So you will find products in the supermarket or the pharmacy that say a probiotic. And but the reason that EFSA got established is to protect consumers against the bad products that are out there. And I can tell several anecdotes about products that are indeed bad, that contain a pathogen, which is definitely not what you want.
That's not a good idea.
No, where the label says that it contains 1 billion microbes of microbe A, and it appears to be microbe B. Or that it says there's 1 billion microbes A, and it's not 1 billion, but only a handful. So, yeah, maybe
wow.
So there's a lot of products out there that unfortunately do not have the proper quality.
Right.
And the good probiotic producers actually suffer from that, because there are also a number of very, very good products that actually help you in what I call the stool transit or skin allergy, to reduce the number of days that you have the flu or something like that.
Oh,
and to make that link to corona, that may also work.
Send me the link, Koen, of those. I'm really curious.
Okay, yeah, I will.
And obviously, there's still a lot to be found. I mean, you probably have only just started this searching for probiotics.
So as I mentioned in the beginning, most of these microorganisms can still not be cultured as pure isolates in the lab, and with the emphasis of still cannot be cultured, because if we do a little bit of effort, I think it is possible. But now with the molecular tools that we have nowadays, we want an answer in 4 hours. And we don't want to wait 4 weeks for a colony to appear on an agar plate.
Right.
But so I think it is possible, and if we do that effort, then we really learn much more about the inhabitants of our guts and how they interact with one another, and how we can make use of those also for our own benefit. So some of the less easily culturable microorganisms are currently being explored as what we call next generation probiotics. Bugs with exotic names like Akkermansia or Faecalibacterium or Eubacterium and things like that.
Mark, didn't you mention that the wastewater treatment and the professors we had, they actually are working with multidiverse organisms.
Yes. We do the same. That's very interesting because there's an obvious link to things, environmental use of bacteria. For instance, we grow dechlorinating bacteria in our laboratory. We bring them in the soil together with the carbon source to degrade chlorinated solvents, and we've done that for about 20 years now in the Netherlands with great success. So it really helps to detoxify, to get rid of your environmental problem in that way. So that's exactly the same as what you're doing with probiotics to enhance your flora, and also these are not grown in pure culture. No.
Mark, that's interesting because Koen, in one of your presentations, and I think I discovered something that is also linked to chlorine. There are bacteria who digest chlorine or break it apart. But could that also be like a probiotics, because we digest a lot of microplastics, which is a sort of a chlorine kind of molecule?
Essentially, these microbes, if you can find the right ones, they can break down anything that you can get your hands on. I used to say that your gut microbiota will degrade everything that we eat except cellulose. And I was wrong about the cellulose. They can also do that. So not in everybody though, and I still have a theory about that this happens in farmers that get exposed to the microbiota of their cattle. And then, they get exposed to cellulose degrading microorganisms that are present in cattle. So not everybody has cellulose degrading bacteria. That's my hypothesis. I mean, I can't prove it. I don't want to investigate it. I want to have a nice anecdote to be able to tell during a podcast or whatever,
But I would always say that fungi are good at breaking down cellulose and lignin.
Yes, they are. Yes, yeah. So, and we also have fungi in our guts. The usual suspect is Saccharomyces, which is used for bread baking and beer production. So when we eat those products, we also ingest some of those fungi, but also Candida, which is usually known to be a pathogenic fungus. But in your gut, it is not really considered to be that dramatic or that bad. Really under certain conditions where it is allowed to overgrow, then problems occur. But that's the same with microorganisms. If you take a course of antibiotics, then a particular microorganism that that used to be called Clostridium difficile, and most people know it by that name, but Clostridium has been changed recently, but never mind. That overgrows and produces a toxin, and you get sick from that, and you get diarrhea, antibiotic-associated diarrhea. So really, if you disturb that ecology, that friendly population of microbes that coexist there, this is when you start to get issues.
And that's why the Germans give you a probiotic after you've done your antibiotic.
Yeah.
Probably, yes. I somewhere read from if you use antibiotics, your gut you can have gut problems for several years even.
Yes. Yeah. My personal experience is 9 months.
Really?
Oh, wow.
Usually people say between 3 and 6 months you can still see effects on your gut microbiota. Wow. If you're unlucky, it's an everlasting problem.
Right.
Oh wow.
Yeah. And this is one of the reasons why they are now investigating, let's call it poop transplantations, to give those people back the healthy microbiota that got disturbed in themselves. So.
But that goes through the top or through the bottom?
Can be both ways. Mostly it's done through the top, but then through a tube that is inserted past your stomach, so you don't really taste it. But also can be the other way, using a clisma. Yes.
But still, it's quite remarkable, because in water quality, for instance, in sanitation and hygiene, the fecal bacteria are thought to be the most dangerous. I mean,
Yes.
sewage is also quite a dangerous substance to work with. So
this is why you should never do this at home. And you can find YouTube movies of people taking a fecal donation.
Yeah.
I'd like to call it a fecal donation. My colleagues just call it shit. But if you take a fecal donation and blend it with some, I don't know, milk or water and then drink it, that's not the way you want to do that. No, you want to have that done by a specialist in a hospital. So please don't try it at home.
It's quite dangerous, yes.
It's quite dangerous, yes. Yeah, those donations have to be screened for pathogens. They have to be screened for viruses and things like that. So yes.
That's the because in every pool you have pathogens and viruses, but your pool is the breakdown of everything. So it's all those who like to break things down tend to be in there, of course.
The stuff. Is it also true that the bacteria you want to get rid of end up in your pool, or is that not the case?
No, what's in your poo is a pretty good reflection of what's in your gut.
Okay.
Yeah.
It's just they sort of flow out.
I was going to say that. Yeah, they just there's a flow from top to bottom, if you like. Yes.
Yeah.
And what is your darling? Which bacteria is your darling?
We've started to grow an interest in this Faecalibacterium prausnitzii microbe, which is anti-inflammatory, so maybe good for people that have inflammation in the gut. It produces butyrate, which we discussed is very healthy. It's able to degrade a number of dietary fibers, which we explained is healthy as well. So we've started to take it all apart, to look at all of the genes that are present in the genome to see if we can predict how we can increase the population of this particular bacterium in our gut. So, yes.
Super.
But then, while you're talking, do you then also have do we also have different strains of the bacteria? I mean, I know, for instance, like Lanzatech is a company doing gas fermentation, and they found a methane-liking bacteria in the gut of a rabbit, and then they did natural selection to find the one that actually liked to do gas fermentation. And do we humans have the same? On the highest level, it's the same species, but if you go down, to how it's mutated, and then we all have different species.
We all have different species, and that's the level that is not yet being explored, or certainly not enough. And that simply is because of analytical limitations. We can't really explore it to the level of the strain. We're stuck with species and sometimes even with genus level information. So we need a new revolution in analytical techniques to allow us to get to that level, because I'm convinced that at that level it also makes a difference. Yes.
And then genetics are not enough, probably, Koen.
It's a start, right? Because through genetics we can actually identify differences in strains.
Yes.
But to be able to do that high throughput and to be able to distinguish different strains is still a big issue. Yes.
Yeah, it is. Yeah, you have to construct the whole genome of every bacteria.
Correct. Yes.
And that's a heck of a lot of computer effort.
Well, with 100 times a million microbes in your gut, that's a whole lot of DNA.
Yeah.
And the problem is they are not there in equal amounts, right? Let's just say, for sake of discussion, that there are some bacteria that are there by the thousands and others by the millions, and again others by the trillions. So that difference in orders of magnitude of amounts being present also limits what we are capable of doing.
You're sequencing the same genome for a billion times before you get the other one, which are
interesting. Yes, yeah, that's true. Yes.
Yeah. Oh yeah, that's horrible.
But that means there is still enough to be done and to
Yes.
be
Oh, yeah.
discovered.
Yes.
And
Wonderful.
I mean, and to conclude, there is a group of people, I think they call themselves transhumanism. They believe we can take the brain out of the human and then transplant that to a computer.
Oh no.
But if you understand that the brain is connected to our gut, you need to take your gut and the brain together, and that is something biology and neurons. Neurons are chemistry, and our gut system is biology, and suddenly it becomes much more complicated to do so.
It does. Yes.
You cannot just take out some parts to keep everything functioning.
Like Elon Musk moving to Neuralink to connect his brains to a machine, but then you still need your indigenous system to actually power your neurons.
Yes.
Which makes it quite interesting. Yeah, Mark, our gut system is complex.
Yeah, and very interesting. We're through with our time actually, so.
Oh, it flew by.
Yeah.
Okay, then we finalize it. Thank you, Koen, for
You're
being
welcome.
here. And we, at least for myself, I enjoyed it very much to explore a completely different area what we are used to in our previous conversations. And yet again, there's so much more overlap than you would imagine.
Yeah.
And health and bacteria, that is, I think, also the topic of the current state of our planet. The more bacteria that we should create, the healthier we can get. So that's a good starting point also for the future.
Good bacteria.
Yeah, like good bacteria.
Yeah, good bacteria. Yes.
So thank you very much, and for the listeners, hope to see you next time on the Master Beautiful Achievements special series. Bye bye.